Hello class, today we're going to be looking at Chapter 10, Section 4, which is protein synthesis. If you're in your notes packet, it's just the next page in your notes packet, which is page 3, the front of page 3. And we're going to have to kind of skip around in this section of the notes packet to fill everything in. So if you just listen as you go, I'll kind of direct you on where to go to fill in certain things. So the very first thing is called the central dogma, and that's what protein synthesis, or this process of DNA to RNA to protein, is called. And DNA, there's a couple reasons we have to do this. DNA cannot leave the nucleus, and so it will make a copy of itself in RNA to be able to travel to the ribosome where proteins are made. So protein synthesis, if you actually skip in your notes packet to page 4, underneath the triangle where it says products of transcription, there is a place that you can write in protein synthesis definition. So the making of proteins at the ribosome is what protein synthesis is. The amount and kind of proteins produced in a cell determine its structure and function. So every cell will produce different types of proteins in order to, based on what its job is, in order to complete its job. And proteins will carry out the genetic instruction in DNA. There are two parts to protein synthesis, which we'll get to, transcription and translation. And that is basically copying the DNA and then making the protein. So protein review is right next to that spot at protein synthesis on that same page, page 4 of your notes packet. And a monomer is amino acids, a monomer of proteins. So this goes back to chapter 3 when we discussed these things in the biochemistry section. There are 20 different types of amino acids. These are linked together by peptide bonds. And this sequence of amino acids will determine the actual structure and function of the protein. So all the different variations of how you can put these amino acids together determine what the protein will be able to do. So protein synthesis has two parts, transcription and translation. And actually now if you go back to the page 3, front of page 3 under the central dogma, there's a place to put in transcription and translation. So transcription makes an RNA molecule complementary to a portion of DNA. When we say complementary, that means that if there's a then there has to be a complement letter going across from it. If there's a C, then there's the complement letter of G going across from it. So transcription, the way I like to try and describe it is script is sometimes helpful to note in here. And script, if we think about what that is, it's just a writing or a copy. So if you scribe something, you copy something down. And that's what's happening here. RNA is making a copy of DNA. So RNA makes that copy of DNA. Translation is when the RNA is read, and this happens at the ribosome. So first, let's do this. Transcription occurs, that's a U, yep, at the nucleus, because DNA can't leave the nucleus. And translation occurs at the ribosome, where proteins are made. We learned about ribosomes back in chapter 4. So in translation, the way I like to describe it is just like if you are taking Spanish class or German or something like that, you have to take the language and translate it into English so that you understand it. Well, RNA is basically doing that job. RNA goes to the ribosome, and the ribosome helps to translate what the RNA has in the sequencing into an amino acid, which will then produce a protein. So transcription is copying the information down, and translation is turning it into a usable language, or the protein. So RNA, what is it? This fills in the box just below transcription and translation. RNA stands for ribonucleic acid. It is the second type of nucleic acid. The first was DNA. The monomer is a nucleotide, just like in DNA, but it's made up of a different sugar. So RNA ribonucleic acid is made up of ribose sugar, and that sugar is here. And then it's made up of a phosphate group and one of 4 nitrogen bases. So these two things are the same as DNA as far as what they're made up of. In here we do have one difference in the base as far as the 4 types of bases, and then here we have this difference of ribose. I would suggest maybe going ahead and drawing in one of these again, just so you have it written in here, understanding that that is what it looks like still when we're looking at RNA. So our nitrogen bases, you have a spot to put this in as well. We have adenine, guanine, cytosine. These are the same as DNA. And then we have another one called uracil. And so uracil here is the U, and it replaces thymine. So the base pairing rules are now AU and GC. GC is still the same, but this U took the place of the T. So our main purpose of RNA is to transfer genetic material from the DNA, which is inside the nucleus and can't leave the nucleus, to the site of protein synthesis, which happens at the ribosome. So our next little part, how does DNA differ from RNA? And we actually, on your notes packet, there's a space for 3, but I'm actually going to put in a 5th one even. So on here we have 4, and we're going to add in a 5th. So the first, there's different sugars. DNA has deoxyribose. RNA has ribose, sugar. There's different nitrogen bases. DNA has thymine. RNA has uracil. There's different shapes. DNA is double helix. RNA is a single strand. There's different locations. DNA is in the nucleus only, and RNA will go from the nucleus to the cytoplasm. More specifically, it goes to the ribosome. Okay, and then actually I'll add a couple other ones in here. Let's do this. DNA has one version, and RNA has 3 types. We're going to go through those 3 types, but currently just knowing that they have 3 types. And you can see those down below. We're going to go through the 3 types of RNA on your notes packet. Okay, so our 3 types here. Messenger RNA, which is labeled as mRNA, carries genetic information from the nucleus to the cytoplasm. It generally, if you want to put in a little picture, it generally just is a single long strand. And what it'll do is this copies the DNA and carries it to the ribosome. Transfer RNA will carry specific amino acids to the ribosome to build the protein. So transfer RNA kind of looks like a T, and it will carry an amino acid. And on the other end here we have 3 letters, 3 bases, and they're called anticodons. Ribosomal RNA is rRNA, and it's the major part that makes up the ribosome. It makes up that organelle. It's the place where protein synthesis happens, and it's the most abundant of the types. And it generally just looks like a big glob. And what will end up happening is the RNA will travel through the ribosome to get read. We'll go back to that picture. You can kind of see what those look like. And if you want, you can draw those in just to help out with understanding what they are. So how is RNA made? You have a place to put in these instructions. And so we'll write these in the steps. I'll first give you what transcription is again. You've already written this definition in, so if you need to, you can go back and put it in again. The process by which RNA is copied from DNA to the nucleus. So again, a helpful tool, think of the word script in transcription, and the script is the actual original words, or scribe copies the actual words from the original. So we're going to go through the steps here. Steps of transcription. RNA polymerase will bind to the promoter site, which is a section of DNA that says, hey, start here. That's step 1. DNA will unwind and separate. This RNA polymerase will start adding nucleotides to the DNA template strand. This is very similar to when we copied DNA, when we did DNA replication, although now it's called RNA polymerase instead of DNA polymerase. And then step 4, the process ends once the RNA polymerase reaches the termination signal, which just says stop here. Once that RNA is made, it will travel out to the ribosome. It'll leave the nuclear pore and travel out to the ribosome to start doing its job out there. So some definitions here. RNA polymerase is an enzyme used to make an RNA polymer from DNA. And it's just making these new nucleotide bases. A promoter is a starting point on the DNA. The DNA template is a strand of DNA that RNA is complementary to. So only one side of the DNA is copied to produce these proteins. And the termination signal is just the ending point on the DNA. Okay, so this is kind of what it looks like. RNA polymerase comes in, starts reading. We have the direction of transcription being shown there. Our growing molecule, this is our messenger RNA here. And RNA polymerase will leave when it hits a termination signal, and then that RNA molecule is released. So our products of transcription are messenger RNA, transfer RNA, and ribosomal RNA. So our last part here, these products then will move out of the nucleus. They go to the cytoplasm to be used in making proteins. And this last part is going to be the translation part. So in translation, we are making polypeptides or proteins from the nucleotide sequence. Basically, what we're doing is translating one language to another language from nucleotides to amino acids, and the amino acids then are usable. The nucleotides are not really usable for anything, but the amino acids linked together are going to be used as a protein for the body. So our steps in translation. Number 1, mRNA codons move through the ribosome. Transfer RNAs add specific amino acids to the growing polypeptide chain. And I'll show you some pictures of these in just a minute. 2, the process starts at the start codon. It will always start here. It will never start anywhere else. If AUG is messed up, it will not start at all. So it always starts at that 3-letter code. And the process continues until a stop codon is reached. And there's 3 different stop codons I'll give you on another slide. At that point, the amino acid chain is done and it's considered to be a protein. The genetic code up at the top of the back page of your notes packet, you can write this in. It's the nucleotide sequence, DNA or RNA, and the amino acid sequence. So the genetic code is actually a big chart, and we'll show you those, and we'll be using those to determine our amino acid sequence. A couple other definitions here. Codons are the combination of 3 messenger RNA. So messenger RNA is always being read in sets of 3, and that would be a codon, and then we would start another codon. They're always read in these sets of 3 letters. Anticodons are from the transfer RNA, and they're going to be read in sets of 3 as well. They're just called anticodons, and so this comes from that transfer RNA. And what will end up happening is these transfer RNAs have amino acids linked to the top, and they will link up with a peptide bond in order to produce this protein. This is showing you translation just in a general picture. I'll show you a little bit better in a second. The types of codons, 64 codons code for 20 different amino acids, so more than one codon codes for an amino acid. We'll show that in the chart in class. The start codon is AUG. It starts the process of translation. AUG codes for methionine, and every protein in your body starts with that amino acid. The stop codons are UAA, UAG, and UGA. This is what the chart will look like. We will read these as first letter. If I had to look up AUG, I'd look up first letter A, and then I'd find the second letter U, so I'd be in this row here, and then I would find G as the third letter, and I'd come across and I would see AUG. If I wanted to look up CAA, I would do first letter C, second letter A, and third letter A, and I'd see CAA codes for glutamine. Another example, if I wanted to look up UCC, I'd start at U, I'd come across to the C, second letter, and I'd find the third letter C, and that codes for serine. Notice any of these third letter ones code for serine. So UCU, UCC, UCA, UCG. And that does happen on some of these.